Okay, I'm gonna go over some stuff regarding the eye. Okay, so the first thing that we need to know is the anatomy. We have the conjunctiva, which is a thin layer around the eye that protects it and usually gets inflamed conjunctivitis. So then we have here the anterior chamber of the eye that lines the front 1/6 of the eye, and it's full of aqueous humor. And we have the iris that consists of circular muscles that can allow the pupil to dilate or contract. Then we have the lens. The lens is actually being held by suspensory ligaments that are in turn next to the ciliary muscles. The ciliary muscles, together with the suspensory ligaments, are part of the ciliary body. The ciliary body is the one that makes the aqueous humor, which is the one over here that allows the structure of the eye to be round. Then we have here the vitreous humor, which provides nutrients to these cells in the back of the eye, as well as maintains shape. Here we have the retina, which lines all of this. The retina is composed of cones and rods. The rods are in the periphery. They're responsible for vision in the dark. The cones see for color. They're responsible for color vision, and they're usually located in a spot, in a dense spot that kind of like goes inward, and it's called the fovea. So the part of the macula is an area near the retina that has a lot of cones, and the fovea is the one with the most cones. Then we have here the choroid. The choroid is what you find in cats, that is reflective, that allows the lights to reflect twice into the retina so that they can see at night. But in us, it's actually dark. That's why it doesn't reflect anything, and we don't use it. It provides blood vessels for the eye. Then here we have the blind spot. This is where the optic nerve goes to. We can't see there. Okay, so the cornea is also in the front, and the cornea allows light to bend in, and the lens also bends it, but bends it further, and focuses it in the back of the retina. Okay. Next we have rhodopsin. So rhodopsin is a broad, light-shaped structure that has a lot of discs on the inside, and the discs have a pigment called they have a molecule called rhodopsin. Rhodopsin lines those discs, and rhodopsin responds to light. Inside of it, there's a molecule called retinol with an L. I forgot. Retinol with an L. It goes from a trans configuration. It goes from a cis configuration. The cis is elongated, as you can see, it has a tail in the back, to a trans configuration. So when it goes to a trans configuration, it becomes elongated and straight, and this induces a conformational change in the rhodopsin that allows transducin to activate what is called cyclic GMP dependent phosphodiesterase that basically breaks down cyclic GMP into GMP. And cyclic GMP is important because it activates sodium channels so that it depolarizes the cell, so it makes it more positive. But when you transform cyclic GMP to GMP, there is no more cyclic GMP that allows for the sodium to go in. Instead, what happens is the sodium channels close and the cell hyperpolarizes and turns off. Okay, this happens in the light transduction reaction. Okay, when the rhodopsin and the rods are turned off, they in turn activate what are called bipolar cells. Bipolar cells, when they turn on, they transduce the signal into ganglia, and then they send those back to the brain, to the lateral geniculate nucleus and the thalamus, back to the occipital lobe, to the striated cortex, which is part of the occipital lobe in the visual field. Okay, so here is a visual field. Okay, so we have the left and the right, left, right. These tracts are called nasal because they're close to the nose, and then these are temporal because they're on the side next to the temporal lobes. Okay, so here we have the eye with the retina in the back, and here we have the brain, we have the occipital lobe, striated cortex, lateral geniculate nucleus, which is part of the thalamus, and we have what's called the optic chiasm. These are where the tracts cross. So the first thing that happens is we need to notice that in the visual field, everything that you see from the left side of the visual field actually comes from the right side of the occipital lobe. Similarly, the right side of the visual field comes from the left side of the occipital lobe. So what happens is, from the left visual field, you go to the right part of the retina, okay? Then it goes to these nasal fibers, tracts, and then they cross at the optic chiasm, okay? All the way down to the right side of the occipital lobe. In other words, the left side comes from the right side of the brain. Similarly, the right side of the visual field goes to the left side of the retina, and from here it crosses the optic chiasm and goes to the right side, to the left side of the occipital lobe. Here we see the similar thing. So we go from right to left, all the way left into the left occipital lobe. Okay, so that's it. If we have a defect, let's say there's something wrong with our optic chiasm. Let me label it. Okay, so let's say there's something wrong here. Okay, then what happens is these, let's just track them. So these over here are defective. I'm gonna color it in black. Okay, same thing with the blue one here. I'm gonna color this one in black. So what happens if you have a defective optic chiasm, or you're unable to cross there, the right side of your right visual field is blocked. You can't see it. You can't see anything from the right side of your right eye. Okay, and then the left side of your left eye is also dark. You can't see anything. So that's what happens when you have a defect in the optic chiasm. The nerves that cross, leading to those sides are defective. So now another point to make is that cones are approximately 7 million, and rods are about 120 million. So definitely we have more rods than cones. Like I said, the majority of rods are on the periphery. Okay, so they're here on the periphery. And the majority of cones, which are responsible for color, so C for color, are mostly found in the fovea, highly concentrated in the fovea. Okay, you still see sprinkles of them here, but the main point is that they're usually found in the fovea and macula. So there are 3 types of cones. We have red, green, and blue. There's no yellow. Okay. So how can you remember that rods are for the dark? So think of R as in really dark and C for color. And that's pretty much it.